Does size matter? A question older than time itself, but we're going to answer it using fish. The study of fish behaviour is vital to our understanding of how fish respond to a fluctuating environment. The innate behaviours that fish display are often considered hardwired and inflexible, but many fish behaviours are due to learning through experience. Take feeding. Aggressive fighting behaviour can be a huge waste of energy. If small fish can learn to avoid bigger fish during feeding, they will become much more effective foragers. As these interspecific and intraspecific relationships between big and small fish start to establish themselves, fish learn their place at the dinner table. So, does size really matter? We'll find out.
So the current challenge is to further develop the knowledge of the interspecific and intraspecific competitive environment, specifically regarding the advantages or disadvantages behind having a larger sized body and the responses of different fish to this. This area appears to have a research gap, meaning that significant findings could possibly be applied in existing aquacultural practices. With the use of BRUVs, it gives people like marine biologists an effective method for further insight into fish behavior and their community structures. It is said that some marine scientists are encouraging the use of BRUVs as a standardized method for studying fish around the world to increase the consistency among marine literature, as it would allow the comparison of trends of activity or disturbance events through different seasons of the year.
Our aim and hypothesis. Our aim is to investigate if the different body sizes of estuarine fish affect interspecific competition when feeding. Our hypothesis is that it is predicted that larger species of fish would demonstrate increased point events, such as boldness, intimidation, and aggression when feeding in open estuarine environments, while smaller species of fish will exhibit less state events, such as feeding in the presence of larger fish species.
Methodology. The experiment took place over 3 close proximity sites within the Port Hacking estuary in southern Sydney. Site 1, Oak Park, Site 2, Shelley Park, Site 3, Bass and Flinders Point. A portable baited remote underwater video station, otherwise known as BRUVs, was built using PVC piping. A GoPro camera, black mesh bait bag and 2 5-kilogram weights were added to the system. Once on site and prior to entering the water, the black mesh bag was filled with a total of 250 grams of pilchards. A diver entered the water with the apparatus and planted the device 2.5 to 3 meters deep on the sea floor. Once the device was placed and the diver had exited the water, we began a timer for 20 minutes. This time frame was selected to add a buffer zone of 5 minutes either side of the footage. This buffer was to eliminate any potential disturbance caused by the divers. After 20 minutes had elapsed, the diver retrieved the device. This process was repeated at all 3 locations. Analysis of footage. As previously stated, the first and last 5 minutes of footage from each video was not used. This was to minimise any influence the presence of the diver may have had on fish behaviour. Upon reviewing the footage, we became aware of a glaring issue. For some reason, the fish seemed completely uninterested in our BRUV system and eluded our cameras. Sad face. It was then decided, due to time constraints and our apparent poor fishing ability, we were resigned to using footage previously captured by the Australian Institute of Marine Science on a reef system in the Timor Sea. This was chosen as it aligned very closely with our method. 7 minutes of footage from the 3 AIMS videos selected were observed and used to create an ethogram, designed to record the behaviours of the fish. We were restricted to 7 minutes as this was the maximum time the area was undisturbed. Fish behaviours included aggression, avoidance, feeding, curiosity, intimidation with the modifiers passive and active, and finally swimming with the modifiers relaxed and erratic. Boris software was then used to analyse the footage alongside the ethogram using a scan sampling method every 15 seconds, enabling us to construct an activity budget. This process was repeated for the 3 chosen AIMS recordings.
Gaining empirical results to test hypothesis has its challenges. Recording and analysing behaviour and interactions between organisms is a challenging area due to assumptions, bias and subjectiveness. A solution to this is to quantify behaviours and interactions through study of etiology and more specifically implementing Boris technology. The approach taken in Boris was to analyse the Brubs footage by point sampling. This meant all behaviour observed within a 1 metre radius of the bait bag was recorded every 15-second interval. On interpretation of the time budget analysis output from Boris, we found the key findings for Site 1 to be that the mangrove jack was the largest of the species. It was recorded showing the most intimidation and was also found feeding for the largest period of time during sampling. For Site 2, we found the white-tipped reef shark was the largest of the species and again displayed the most intimidation. The dark-tailed sea perch was the smallest of the species here and displayed no feeding during the sampling. Site 3 showed the white-tipped reef shark displaying the most intimidation again. At this location, it was again the largest species. The mangrove jack was the smallest of the species and displayed the most avoidance during sampling.
When observing behaviorograms obtained, there was a notable trend with the results of fish that were larger engaging in more aggressive activities, while smaller fish had engaged in less feeding activities. These results do relate to our hypothesis. These results match ones that have been seen within official works. Works of Works in 1983, having observed a trend of wild which noted that larger sizes were preferred for species, and that aggression was prevalent in the overall goal of obtaining food. This also matches works done in 1994, in which, when observing samples from the same species, it was found that the ones that were smaller in size were comparatively less aggressive to the ones that were larger in size. A potential reason for this correlation of size and aggression could be due to the predator species eating the prey species, while the members of prey species that had managed to survive would ultimately be the ones that were smaller, faster, and just more capable of evading the predator species, and as a result lead to behavioral changes the offspring of the surviving species, members of prey species, being more likely to avoid conflict rather than other species would. This idea is supported by works in 1984, which found that the age and relative size of species had led to alterations in interactions that were being held between the members of certain species. Some of the main limiting factors within this experiment were that the locations picked for sites were relatively close to each other, leading to them having less of a widespread implication overall across the entire environment. The method in which the overall apparatus for recording results being set up in a way that made the overall environment relatively unnatural, which does alter the results somewhat, and that only one size of bait was tested throughout the entire experiment. For future experiments, it would be advisable to try out experiment in different locations to see if that alters the results, try out different methods of gathering the data in the first place, or by trying with different types of bait or quantities of bait. Overall, this could lead to important data being gathered and later on have it be used to form greater conclusions, and be used in methods such as how to construct farms containing multiple species of fish, and how to properly manage certain ecosystems that are in need of management